Okay, thanks for coming. I feel like I should thank you guys because you're still coming, so thank you. Okay. Business. Exam 2 is next Tuesday. I have put this week is review and recitation, and I put something on Blackboard, and it's basically, it's an opportunity to spend 2 hours with me going through everything that we need to go through for the exam. I'm not going to do it myself, but I gave you a list, and you guys will sit in groups and go through it. Feel free to get a head start, Thursday people, and pull that and just start working through that stuff. And then the goal is for you to be able to draw the cell cycle, as in like, G1, S, G2, M, and then put all the different components into that drawing so you can start to kind of have the big picture of what's going on here. So the goal with doing that starting today is that you can have a full week to study. So today's recitation got started, but the rest of you guys feel free to go ahead and get started. Dr. Texel's recitation section, come to Thursday, either of my recitations. Don't bother going to hers because she won't be there. If you have trouble making it to either of those, email me and we'll work something out. Send exam questions by Friday end of day. Remember, multiple choice, must have 4 possible answers. None can be all of the above or none of the above, only I'm allowed to do that. And they all have to be critical thinking questions, no recall. Okay, any questions about any of this stuff? Good. Okay, so this is what we've done so far. It feels like a lot. I don't know. Is it?
Yeah.
Okay. I always feel like I don't teach enough. No. That's what people have said to me before, like peer evaluations. But maybe they're coming up days when I'm just introducing something. I don't know. Anywho, okay, so last time the way we ended it, we've been through all the kind of backgroundy stuff. And so this is a lot of stuff, but I want you to understand it's part of one process. And so the goal of you getting through it all, putting it into one big picture, or chart or something, is to figure out how to put it all together. Because this all happens during the cell cycle, right? All these things we've been talking about are things that regulate or happen during the cell cycle. So after we went through S phase, we kind of finished up with talking a little bit about epigenetic inheritance, right? Inheriting your histones and those modifications. Okay, so now we're going to move on to M phase, mitosis. So here are some highlights, right? So the first thing that you know happens, it's interesting. Why? Oh, it's backwards, huh? I'm looking at it going, why are we starting with telophase? So you're in interphase. Whenever your chromosomes your DNA is not condensed, that's always interphase, right? So you're in interphase, and then you start mitosis, and the first step is condensation of your chromosomes, right? So what cyclin CDK complex do you think activates condensation of your chromosomes? M-cyclin, MCDK, right? So your M-cyclin levels have gone up, bound to your MCDK, activate chromosome condensation, right? So why would you want to condense your chromosomes? Yeah.
Prevent another round of DNA replication, as you already said.
Oh, to prevent another round of DNA replication. That's interesting. Yes, definitely you cannot replicate condensed chromosomes. What else, though, is preventing another round of DNA replication?
Expression of other cyclins, like a cyclin.
Well, so I want you to think about it for a minute. We talked, because you actually led me into an interesting thing to talk talk about here. We talked a little about what regulates DNA replication, right? Yeah.
Oh, I was gonna ask the why, answer the why.
Okay, go with why.
Is it just because of segregation?
So you can have nice, clean chromosome segregation, right? But also, just to go back to the question about DNA replication and how you don't want it to start again after it's done, go into M phase. Why doesn't it just start again? Yeah. So what I'm hearing is there's something about Gemini, something about some complex. You're getting in the right direction. Who wants to expand? Yeah.
Is it the definition of separate and inactivate APC?
Okay, so separase and all that has to do with chromosome segregation, but we're asking why S phase doesn't start up again.
No, because this does a loop which prevent binding of the CK1 to the binding site, doesn't it?
But okay, so you're saying that you're degrading
Yes.
M and S cyclins.
Yes.
Okay, so M and S cyclins are degraded, so you can't turn on S phase. What other thing is going on? Yeah.
I think it was like the whole, because you need to get all those proteins together one more time if they've already been degraded. And if you have not like made them, freshly made them, you can't really have that origin of replication being like all those being present and then
Okay,
remaining.
right. So origin of replication, you need to have that pre-replicative complex set up there. And we've just degraded some of those proteins and inactivated them. And then I heard something about geminin. And so at this point, APC is off. So geminin levels are high and they're inhibiting pre-RC formation. So I like that you kind of led me down that path because yes, chromosome condensation completely prevents S phase. Yes, S cyclin degradation prevents S phase. But more than anything is your origins are not ready. All the components required for loading your helicase, they're not there, so you can't replicate your DNA. All of those things are missing. That's such a great essay question, isn't it? Yeah. Okay, good. Chromosome condensation.
What was
So,
the how again?
huh?
On the last slide, what was the how?
How? Well, we didn't get there.
Oh, okay.
You're right. So how are chromosomes condensed?
No, sorry.
It deals with condensin. Did anyone read about condensins while they're doing their quiz? Did anyone think about condensins? Yeah.
Well, capacity holds two sister chromosomes together, and condensing is kind of trying to restructure it so it's not condensed.
Right. So condensin is what is it? Is it a plant? It's a protein, right? It's a protein, and it acts to kind of bind to the chromosomes and do what to them? Condense them. That helps, right? Want to name proteins by what they do. Yep. So it helps to kind of bind to chromosomes and keep them condensed and compact so that they can segregate cleanly. So with how the activity of condensin and condensin needs to be activated, and it's activated by MCDK. Okay, so this is what it looks like, right? So interphase, you just have, that's your nucleus. Yes?
Do condensin and cohesin, do they work together in this process, or is it just condensin?
Yeah, so I just got the question, does condensin and cohesin work together? No, you're doing that because they're on the same slide. And because they have similar structures. But they actually function similarly in terms of binding to DNA and keeping things together, but totally separately in reality, right? Because condensin is actually binding to histones in the DNA and helping to keep it compact and condensed. And cohesins are binding and keeping sister chromatids together. Yeah, but you can. I think I see where you're getting that from because they're on the same slide. So at prophase, you get the activity of condensins, and that's so you start to see your individual chromosomes. So there's actually a condensin complex, and so the protein, you can see the protein condensin. I think it's a dimer. We'll see that in about one slide, and it's stained here in red, or it looks pink here. Blue is DAPI. This is right out of the textbook, and so you can see it's bound along the chromatin and helping to condense the chromosomes. So two things need to happen, right, for chromosome segregation. First, those chromosomes need to condense, and that's done using condensin. And then you need to have your sister chromatids paired and held together, and that's through the activity of cohesins. And that is why you're thinking of them together, is because if you looked at these slides or you saw last year's lectures, you would have seen that I have them on the same page, which might mislead you to think that they work together. They're both required, but they do different things. So cohesins keep sister chromatids together, and they're cleaved at the onset of anaphase. And they're both made of proteins that are from the same family. And so they have similar domains and similar structural features, but they do different things. So here's another picture. So this is showing another way to look at condensins and cohesins, and it's just where they bind. That's different. So with sister chromatids, you've got these DNA binding domains, and with sister chromatids, they're holding one DNA binding domain is holding one sister chromatid, and the other's holding the other one. That's how it's keeping it together. Whereas condensins are binding along one, just binding along the DNA.
It's double-stranded DNA.
Double-stranded DNA. Yeah, this is not accurate by any means. Yes?
So condensing has nothing to do with two sister chromatids. It's there even after they've separated.
Yes, so if you have, let's say you have condensin still active and bound, and then so the question is, does condensin have anything to do with holding sister chromatids together, right? So let's say you do something to inhibit cohesins, but not affect condensins, your sister chromatids will just migrate apart. Condensins so what condensins are actually doing is they're binding to methylated histones. So histones are all along the DNA, and they're compacting and holding it together that way. And the activity of condensins is regulated by phosphorylation by MCDK. So can you have condensation of your DNA prior to levels of M-cyclin reaching appropriate levels? No, right? Because M-cyclin is required to activate MCDK, and you need that MCDK to phosphorylate condensins so that they can bind the DNA. Yes? Yes, it's an activating phosphorylation. Which kind of makes sense, because if condensins are around in the cell, do you want them binding when they shouldn't be? No. Otherwise, you'd always have cells that looked like prophase and not like interphase. So that difference is due to the activity of condensins. Any other questions? Okay. Oh, so these are cleaved during anaphase. How? Right, separase. Separase. Why would you cleave cohesins at anaphase? You can't segregate your sister chromatids without doing that. Right. And what ubiquitin ligase is involved with that? APC, right. Okay. Okay. So there are actually two models for how cohesin holds sister chromatids together. So either you might have this complex, so it's two different chains of proteins bound together. You'll either have the complex actually just wrapping around, or maybe kind of linking together, like kind of like chain link. Those are different models. You don't need to know, just acknowledge the fact that there are different ways that it could work, and people are still studying that. Well, actually, I should look it up because the way it takes, the length of time it takes between research being done and making it into a textbook is so long that it might be well established at this point. Okay. So most of the cohesins are removed actually during protease. But only the final cohesins are removed by separase during anaphase. So in actuality, you'll have, where's prophase? Here's prophase. You'll have a whole bunch of binding, but it's the activity of separase that's going to remove those final cohesins that are around the centromere during anaphase. And that's through the activity of separase, which is activated by APC indirectly.
Question.
Yes?
So that means cohesin is not just at the centromere, it's all around the...
It's actually yes. So cohesins actually are up and down both sister chromatids, but it's just that the only part that remains in the end prior to anaphase, so at metaphase, is around the centromere. Yep.
Is that part that's so called
Well, so the kinetochore is actually has to do with binding to mitotic spindle. So, and we're going to get to that at the end of this lecture. But kinetochore is a bunch of proteins that assemble on the centromere and directly interact with the microtubules in the mitotic spindle, and that's what allows for chromosome segregation.
So they're originally they're not on that centimeter. It's on later on.
So you're asking about cohesins, right?
Yeah, like I'm asking is connected there and
So the question, so kinetochore is a complex of proteins that bind to the centromere. You're asking when it's there.
yeah.
I want to say, I don't know the exact timing, and I don't know if it only comes at a certain point in time. Let me think for a minute to see if we can reason through this. So, once you're condensed, and then you're going to line up, it's actually the mitotic spindle that lines you up, which means the kinetochore needs to be there that early, right? Because without the mitotic spindle, you don't ever line up during metaphase. So it's at least there that early. I don't know. So the question was basically when does the kinetochore bind to centromere? And it's actually not one protein; it's a complex of proteins. But I think as soon as it's needed, it needs to be there. But I'm not sure how much earlier. Okay. So sister chromatid. That's rated D. Separation is triggered by APC. We just said that. Right. So normally you'll have. So right here in G2, right prior to M. You've got your sister chromatids, right, because you just replicated your DNA. They're bound and held together by cohesins. And then your MCDK is going to activate M phase. So you'll start mitosis, and then later on, APC will become activated and will degrade securin. And securin is an inhibitor of separase. And as soon as that inhibitor is degraded, then separase is active and will cleave the cohesins. And that happens between metaphase and anaphase. Questions? Okay, so why is sister chromatid cohesion so important? Why do we have cohesins keeping sister chromatids together? Okay, so they can be equally pulled apart. Upgrade to Mavericks. Well, not right now. Should I upgrade to Mavericks right now? I feel like the timing is bad. Yeah, so what happens if you don't have cohesins? What's the outcome in terms of your daughter cells? Yes?
You won't progress through the cell cycle.
Well, you won't, so big picture, you won't progress through the cell cycle, yeah.
Like distribution of genetic information. Like one will get extra Down syndrome and all the other.
So your genetic information will not be equally distributed. Give me more. So cohesins hold sister chromatids together. So will the mitotic spindle still be able to bind to kinetophores? Sure, why not? I guess so. But will things segregate in any sort of order? Like will there be any rhyme or reason to what daughter cell gets what chromosome? Probably not, because there's nothing to kind of hold them together and get them organized so they can segregate one sister chromatid to each daughter cell. Right, so that's what happens from metaphase to anaphase. Okay, so you need the mitotic spindle for chromosome segregation to happen. So this is going to be like super quick background about microtubule dynamics. Hopefully you guys learned about it as you were doing your quiz, right? So microtubules, they're made up of tubulin dimers. And they're stabilized by the presence of a GTP cap. Who knows what a GTP cap is when we talk about microtubules? Yep, so GTP, yep, but so what's the purpose specifically of the GTP cap? Or what is the GTP cap? Yes?
Doesn't it stabilize it so that you don't get rapid removal?
Right, so it stabilizes, but where's the GTP bound? Huh? Okay, so I'm seeing that a lot of people need a little refresher. I was hoping my 408 former students would teach this for me, but you're making me do some hard work. Right, so some people have learned this in more detail, other people have not, so I guess we're all back at the same place. So microtubules are made of tubulin dimers. A dimer is two proteins bound together, right? And so it's an alpha tubulin, beta tubulin dimer. And your beta subunit has the ability to bind GTP and hydrolyze it. And GTP bound dimers like to be added and added to microtubules so that the microtubule can grow. GTP bound tubulin dimers, they like to add on to growing microtubules. So microtubules are dynamic things that are constantly growing and shrinking. And it's whether they're growing or shrinking pretty much depends on the concentration of tubulin dimers present. And then there's all these proteins that help to regulate whether they're growing or shrinking. But we're not going to get that detail. But GTP-bound tubulin dimers like to be added to microtubules, so they encourage microtubule growth, microtubule lengthening. And so they get added basically head to tail to head, right? You can think if your head is the beta subunit, which has this pink square, which is GTP in it, and then they call the tail the alpha subunit. It's going to be added so your alpha subunit is binding to the beta subunit on microtubule. And then GDP, so after it's added, the GTP will hydrolyze, okay? And so GDP bound tubule and dimers don't like to bind. They don't bind very strongly to microtubules, and they like to detach. So you can imagine at the front end, so the other thing is there's polarity to microtubules. There's a plus end and a minus end. The plus end is where your dimers add onto, and your minus end is where they tend to leave from. And so when your GTP-bound dimer adds to your microtubules, there's a lag time. It's not like the GTP is going to immediately hydrolyze. It takes a while. And so when you have a ton of tubulin dimers in the cell, then you end up getting this GTP cap, which basically means your plus end of your microtubule is full of tubulin dimers that have GTP, where the GTP hasn't yet become GDP. Okay? And that stabilizes the microtubule. So if you have this GTP cap, then your microtubule that's growing. If in the cell all your tubulin dimers are all busy being bound to microtubules, then there's no free tubulin dimers. Then there's no more ability to add dimers, right? Because they're all used up. Then slowly, the GTP will hydrolyze, and then they're going to want to detach, and then you get this shrinkage of your microtubules. And so that's what we're talking about when I start to talk about microtubule dynamics. It's the fact that they're growing and shrinking, and whether they're growing or shrinking depends on the GTP bound state. Yes?
Is that GDP is what makes them short?
Yeah, so once the GTP is hydrolyzed to become GDP, then the binding's not as strong between the tubulin dimers. And so the subunits are more likely to leave. And that results in shrinkage of your microtubule. Why am I talking about this? Because your mitotic spindle is made of microtubules. And chromosome segregation occurs because you have this rapid disassembly of your microtubules while they're bound to your chromosomes. So it's useful to understand a little bit of what makes them disassemble, what makes them assemble, and all has to do with GTP or GDP bound state. And if it's GTP bound, it's called a GTP cap. So it's not like there's GTP sitting on top of the microtubule. It's just that all those tubulin dimers are GTP bound. Okay? Questions about this? Yes?
Is that the positive part?
Yes. Yeah. So, and I'm going to show you guys a nice picture about how that relates to chromosome segregation. Okay. So, right, so there's this concept of dynamic instability, which means that GTP hydrolysis is going to promote what's called catastrophe. Catastrophe is when you lose your GTP cap, and then you get this rapid disassembly, which looks like this. And rescue is when suddenly you've got all these monomers dimers available, and they can add again, and you can get growth. So that's this dynamic instability. Okay. So your centrosome is the microtubule organizing center of your cell. That it's kind of like the sphere full of microtubules radiating out in every direction. And normally, when you're in interphase, you've got this centrosome, that's hanging out by the nucleus, and your microtubules are radiating out in every direction. We have no paper today. Sometimes people leave me paper. Oh, sure, then I can, yeah, then I can use that.
That's super zoom.
Oh my lord. I look like I'm an old lady. Look at that. Woo! You can see my skin cells now. Who needs microscopes? Okay, there. Let's see how that looks. Okay, so during interphase, your centrosome is like right by your nucleus, kind of, or actually, let's make it over here just for kicks. And you've got your centrosome is kind of covered with these proteins called gamma tubulin ring complexes. Don't worry about that detail. That basically help those microtubules to grow. And so they kind of coat the whole entire cell, which is why every single time I show you immunofluorescence of microtubules, you can always tell where you are in cell cycle, because interphase, it's just kind of these lines all over the cell. And at this point, they're just really being used for movement of vesicles and organelles and stuff like that. And these motor proteins walk along carrying things. But in M phase, you get duplication of your, so cross that out, by the way, that's not really there. In M phase, you get duplication of your centrosome. And what does it become? Right, your mitotic spindle. And so that's why you can start, and I notice the nuclear envelope is gone, because that happens too, but we're not going to worry about it. So oops. So the centrosome is where all of your microtubules are radiating out from, and it needs to duplicate in order to form your mitotic spindle. So here's a bipolar spindle, bi-2 poles. You could see centrosome 1, centrosome 2. The green are microtubules radiating out. They always have microtubules radiating out. And then here it's showing some microtubules that are pushing out against the plasma membrane. If you could see the plasma membrane, it will kind of be circled around here. And that's how they center. And then you've got some that are interacting with your chromosomes and some that are interacting with each other. This is a multipolar spindle. We're not going to worry about that. So you've got your mitotic spindle, and then you've got these motor proteins that are helping to position the mitotic spindle. Right? So motor proteins have two main sides, your head and your tail. Your head tends to bind to cargo, whatever it's moving. And the tail, usually they have these motor domains that walk along microtubules, literally walking along microtubules. And so this can help to, so during interphase, this is what it's doing, what you see in A. It's literally binding to these vesicles and walking along your microtubules to move things within the cell. But during mitosis, sometimes your head will actually be interacting with the plasma membrane or the cell cortex, and that's helping to position the mitotic spindle to where it needs to be. And then you also have different types of motor proteins. These are mostly kinesins and dyneins. So you have different types of motor proteins, and some are going to have two different motor domains, and they're going to walk in opposite, well, in the same direction, but really opposite direction because they're anti-parallel, and help to slide microtubules against each other, again, to position the mitotic spindle. So this right here is showing your mitotic spindle. You've got your centrosomes, you've got microtubules, you've got these astral microtubules that are going to be pushing against the cell cortex, and they're going to be bound by motor proteins that walk along the microtubules but are also bound to the cell cortex. And so they're going to be helping to move that mitotic spindle and put it in place. Then you have these other motor proteins that are going to bind to microtubules from both poles and also help to center that mitotic spindle. It's really important that the mitotic spindle end up at the right place, right? Yes. Microtubules?
Are sticking off the centromere itself?
Centrosome.
But they're created at that region as well.
Yeah, so I'm trying not to overload you with microtubule dynamics, but in the process, probably confusing you. So if you go back to this, so that's your centrosome, okay? Don't confuse centrosome with centromere. Centromere is that part on your chromosome where your kinetochore binds and mitotic spindle is going to bind and segregate, right? Different. Centrosome, I know, not useful to make them sound the same. Centrosome is your microtubule organizing complex. It is made of centrioles and gamma-tubulin ring complexes and microtubules, and that's essentially, you can think of it as like a seed where multiple microtubules grow from. And so that's duplicated and becomes the bipolar spindle. Okay? Yes?
What determines how many centrosomes we have, and does that number change during the cycle?
What determines how many centrosomes we have and does it change during cell cycle? The number of centrosomes you have is determined by how many your cell normally has. So normally you have one per cell. In mammalian cells, as far as I know, you have one per cell, and it duplicates through cell cycle. So you just get duplication. So there's two, and that's why you have a bipolar spindle, because you're only going to separate into two cells. I think maybe during development it's slightly different, and in different cell types it's slightly different, but just the standard mitotic spindle, you have one centrosome per cell, and it duplicates early in mitosis. Yes?
So this is kind of like the middle of the mitotic spindle, right? The two
Your poles.
poles. Like
Yeah.
that's in
So
the-
this duplicates, and that becomes your two poles. So don't worry about this multipolar spindle. Focus on your bipolar spindle, and that each yellow dot is one centrosome. So it's duplicated early in mitosis, and that forms your spindle in prophase. Yeah.
You said that the head interacts through mitosis. The head interacts with what position?
So for your motor proteins, during mitosis, it depends on the motor protein. If you're a motor protein with a head and a motor domain, your motor domain always walks, or tail, your motor domain always walks along your mitotic spindle, and your head always interacts with some sort of cargo. That cargo in mitosis might be the cell cortex, which is basically your plasma membrane. And so it kind of makes sense when you consider you're trying to separate and position your mitotic spindle appropriately. And so having it interact with your cortex and then walk along your microtubules of your mitotic spindle, it's going to pull your spindle outward. And then if you have this type of motor protein that has two motor domains, they're going to walk each to the plus end of your microtubule, and that's going to slide them against each other. And that also helps to position your microtubule. Your mitotic spindle. Yes? How can it go in two directions at the same time? Are you talking about the one with the two motor domains? They both walk to the plus end. So you've got a protein which has two motor domains. Both go to the plus end, which is the part of your microtubule where your tubule and subunits are added. And so since this one is walking this way and this one is walking this way, it actually, what it does is your motor protein's not moving. What's happening is your microtubules are sliding. It's going like. And so since they're sliding, it's pulling that centrosome. So what you have is pushing and pulling going on that positions your mitotic spindle correctly, so that when you have chromosome segregation, right? Because you can imagine if your mitotic spindle is in position correctly, and you've got this one here, and then this one is like right over here. Like you need to have some sort of plane for segregating your chromosomes, so that when you have your daughter cells, everything is equal and appropriate. Yes.
So two subunits are present inside the centrosome as well as outside.
So let's go back and let's go back to our microtubules. Your centrosome, think of it as a place where microtubules grow from. Okay, and each microtubule is going to start. It's almost like its root is in your centrosome, and it grows outward. So tubule and subunits add to the plus end, which is the part that's furthest from the centrosome. Okay. Questions? Okay. So now we've duplicated our centrosomes. We've moved so we have a bipolar spindle that's in the appropriate place, right? So the microtubules of the mitotic spindle need to interact with the chromosomes so that you can segregate. And so that's through the kinetochore. And so you've got your chromosome, and then your kinetochore is going to interact with your centromere. And your kinetochore is where your microtubules bind. And so this assures that there's a location for your mitotic spindle to interact, and allows for chromosome attachment to the spindle and proper segregation. This complex actually can be phosphorylated by Aurora B kinase if things aren't happening quite right. Because you want your mitotic spindle to attach appropriately to the sister chromatids. That means one microtubule from one pole, one microtubule from the other pole, attached to one of each sister chromatids. If it doesn't attach correctly, it needs to detach, and there needs to be a do-over. And Aurora B kinase is available to bind and phosphorylate the kinetochore and detach the mitotic spindle if necessary. It actually senses the tension. I think I go through that in a little bit more detail. Okay, so let's go over the kinetochore for a minute. So kinetochore is not one protein, it's a complex of proteins. And this is an experiment where they identified the proteins that make up the kinetochore. So if I told you that you know kinetochore binds to centromeres, and you already know SEMP-A is the histone variant that's present in centromeres, how would you go about identifying kinetochore proteins? So you guys can look at this, or you can discuss with your friends, but I'm going to give you guys a minute to just think about, if you had to identify the proteins that make up the kinetochore, and you knew that the kinetochore assembled at Sempe, what would you do? How would you do it? Discuss.
Figure out
I know, I'm like, what we have in it. I know that
Your goal is to find, the thing is you have central mirrors on both sides. Kind of play that slide right next to central mirrors. Then you have these like two little like big price standards. Yeah.
Oh, really? Oh, so you
Because if you knock out central mirror, there is no easy to separate. He's saying knock out central mirror, but the thing is they don't have Sunday.
Yeah, it doesn't matter which one you knock out something.
Because if you
You can knock out, like, shut it off.
I mean, you knock out production of that protein, so it knocks out the deep.
Basically, you silence the protein. Turn it off.
So if you, he's saying like if you knock out central mirror,
Oh, no.
now it's central mirror like
To knock out something.
Maybe he's just joking.
Okay, there's too much silence in here today. Okay, so if you were trying to identify kinetochore proteins, and you know the kinetochore sets up interacting somehow with Sempe, what would you do? Go ahead. You have an idea.
I have an idea, I just don't know if it's right.
It doesn't matter. It's okay.
So can we tag SIMP-A and then isolate SIMP-A to see what proteins are associated with it?
Exactly. So you can isolate Sempe and see what binds to it. That's exactly what they did here. So you express Sempe in mammalian cells because that's what you're studying. And then you isolate it by immunoprecipitating it or just isolating it by some tag that you put on it. And then you see what it interacts with. And you can identify interacting proteins through mass spectrometry. Does anyone know what mass spec is? Has anyone learned about it before? Chemistry. Yeah, basically what it does is you can determine the sequence of a protein or peptides. So what you do is you fragment your proteins into peptides. Peptides are short, like 10 amino acid long fragments. And then based on their basically molecular weight, you can figure out their sequence. And then you blast it against all known protein sequences. And if you have a certain number of peptides that match certain sequences in the database, then you can say this protein was here. So usually what you do is you'll tag and you'll express whatever protein of interest. You'll use that protein to pull down its normal interacting partners. And then you'll and before you could do two things. One, you could do old school where you run it on a gel and you cut out individual bands. And so that's what this is showing. It's a silver stain and they showed all these different bands. You can cut out bands that you're interested in, but now, actually what you can do is you can take the whole mass and send it away, and they'll mass spec the whole mass of stuff that came down. And so they found a whole bunch of different proteins that are associated, a bunch of SEMPs, some histones, NPM1. But basically, this is how you identify protein complexes. Questions? Yeah.
But if you tag in SNP, you are technically just figuring out that it binds to the centromere. Like, okay, I understand that SNP binds to centromere and centromere binds to the kinetochore, but by targeting SNP, you're just proving that it binds to the centromeres. So how would you distinguish that a protein would be part of the kinetochore?
Well, so what you do is, so you're saying, well, you know what binds to centromeres, aren't you just going to find centromeres? Right. Well, centromeres, what are centromeres, right? Centromere is a DNA sequence that tends to associate with SNP, right?
Mm-hmm.
So if you pull down SNP, you're going to find all the other proteins that are bound to SNP. If you purify SNP from cells, you're going to find all the proteins that interact with SNP. And you can expect, since you know that probably the centromere is somehow involved with kinetochore and somehow involved with chromosome segregation, the identity of those proteins, the ones that you identify are most likely to be part of your kinetochore complex.
But that's what I'm saying. How would you know it's part of the kinetochore, not the centromere?
Well, because you know what the centromere is already.
Oh.
Right? So if you know your centromere is made of SNP-A, and so it actually they did find a bunch of things that they know. They found SNP-A, SNP-H, and they found a whole bunch of different things. And so then you have to use your knowledge that you already have and say, well, we already knew it should bind to this. We already knew, oh, but there are these 5 novel proteins that came down that we didn't expect to see.
So it's like an elimination process.
It's an elimination process. I did this before where we were trying to find a protein complex. And we knew a bunch of proteins that should interact, but we were trying to find other proteins. And so when we ran our gel, we were able to say, oh, well, this is good. We know we're pulling down what we wanted because we're seeing what we would expect to see, plus we're seeing new proteins that we don't know about, and those are the ones we want to identify. Yeah, good question. So just because you identify some proteins interacting with SNP doesn't mean it's a kinetochore, right? So they had to actually prove that it was behaving the way that these proteins are behaving the way they should. So they found a whole bunch of these different proteins. This ACA, for example, this is anti-centromere antibodies. So that is just identifying your centromere. And then they found this SEMP-N, and so they wanted to know whether or not it's going to localize to the correct location. Because just because you purify something from cells doesn't mean it's doing what you think it does. So they did immunofluorescence, and they're comparing anaphase and metaphase. And then they're looking at whether or not SEMP-N, which is this kinetochore protein that they think is a kinetochore protein, they're looking to see whether or not it's in the same place as the centromere, because you would expect it would be. And then they're staining also for DAPI. And so they're seeing SEMP-N and the centromere in the same place, and then they overlap it with the chromosomes, and it's exactly where you would think it would be for metaphase, right? Because you have all your sister chromatids lined up. And then at anaphase, they should be separating, and then you see it separating at anaphase. So that gives you extra confirmation that it's a kinetochore protein or some protein involved with chromosome segregation because it is present where you would expect it to be. And then the next thing you could always do is get rid of that protein and see what happens, right? Because if you know your kinetochore is where your mitotic spindle binds, then if you get rid of proteins in your kinetochore, Should you have normal chromosome segregation? No, and so that's the other thing they did, right? So normally you should have your mitotic spindle binding and segregating your chromosomes, right? If you get rid of this sump end that they were studying as part of this kinetochore complex, do you see normal chromosome segregation here? You can see there's chromosomes that actually never bound, which means that something was wrong with that kinetochore. They're not all lined up properly, they're not segregating properly, there are kind of errant chromosomes sitting around. Any questions about this and how they did this? So you would expect proteins that are part of the kinetochore to localize to the centromere. That's what they did, and you'd expect that they would be required for attachment of chromosomes to the mitotic spindle. Another thing that they showed. Okay. We've got 15 minutes left. So let's see. Do we want to keep going? You guys are like looking like dead people today. So we've got about 10 slides left. So you have a choice. We could power through, finish. You can go home, listen to it, digest it, ask questions on Thursday, and we can use Thursday for review. Or we can spend the next 15 minutes with you guys asking whatever questions you have, finish these 10 slides on Thursday, and then use the rest for review. What do you want to do? Power through? Yeah? Okay. Okay. I'm just making sure, because when I see a lot of this, I think that I'm like not being effective, and I want to stop. Okay. So we did a paper last week on spindle checkpoint, right? Last week? No. 2 weeks ago. Right. Right. I don't even know. Right. Back in the winter. Not like the spring, beautiful spring day we have today after spring break. So we learned that spindle checkpoint gets triggered when the mitotic spindle does not attach properly, right? Okay. So mitotic spindle needs to attach appropriately to the chromosomes, otherwise all sorts of things go bad. And so there's a way to regulate this, right? So you need correct attachment of the mitotic spindle to the kinetochore, otherwise you can't progress to anaphase. Otherwise, spindle checkpoint is activated. And so correct attachment means one mitotic spindle is going to attach to one sister chromatid, the other one pole is going to attach to the other sister chromatid, and then you can have appropriate segregation. You can have incorrect attachments. What if only one pole attaches to both, right? So that's what's showing in number 1. Or what if one completely doesn't get bound to it all? And so there are ways to regulate this or to check this in the cell. And that has to do with sensing the tension between, basically the tension in the mitotic spindle. If you have appropriate attachment, the tension is correct. And if you have this inappropriate attachment, it will not be right. And it will be sensed. And that's where Aurora B kinase comes in. So if you have an incorrect attachment, such as, what one pole attaching to both sister chromatids, then Aurora B kinase will phosphorylate the kinetochore proteins, and that allows the microtubules to detach, and it allows for an opportunity to reattach correctly. And if you have continuous incorrect attachment, then you get activation of the spindle checkpoint. So if you knock out Aurora B kinase and you prevent it from working, then you're going to get improper chromosome segregation, because you're going to get incorrect attachment, and then 2 sister chromatids might go in one direction, 2 sister chromatids might go in another, one might get left behind, who knows? Okay, so that's Aurora B kinase. It's a way to detach the mitotic spindle if it incorrectly attaches. Yes?
So if Aurora B kinase is functioning properly, the spindle checkpoint is inactivated,
Right, so this is a question that came up in recitation today. So aurora B kinase is, if it's functioning correctly, then it allows a redo of mitotic spindle binding to kinetochore. But maybe something's wrong, and it, despite a number of redos, it doesn't attach correctly. That's when MAD2 and all that come in, and then you get activation of your spindle checkpoint. So it's like double assurance. So you get a number of redos, and then MAD2 and BUB, and all these proteins that we spoke about in recitation 2 weeks ago, those proteins activate the spindle checkpoint and prevent chromosome segregation. Okay, so I'm saying aurora B prevents spindle attachment. How does that happen? What does aurora B do? It's a kinase. What does it do? It phosphorylase, right? So phosphorylase your kinetochore, and that's going to inhibit or decrease binding between the kinetochore and the mitotic spindle. So it allows for it to detach. Okay, so if the mitotic spindle does not attach correctly to the kinetochore, then you get spindle checkpoint activation, and that's through the activity of MAD and BUB and these proteins we spoke about 2 weeks ago. And spindle checkpoint activation, what happens if you activate the spindle checkpoint? What happens in terms of cell cycle? You can't exit mitosis. What else can't you do? Anaphase, okay, right. So basically you can't segregate your, you can't move to anaphase. You can't segregate your chromosomes. And what happens is you activate, you actually inactivate or prevent activation of APC by inhibiting CDC20. So CDC20 can't bind APC, can't activate APC, and then all the things that APC is supposed to do can't be done, right? So APC is supposed to do what? Degrade securin, so you can't get cohesin cleavage, you can't get cyclin degradation, all these sorts of things that need to happen to go from metaphase to anaphase cannot happen. If you have an issue with your spindle checkpoint, then that's seen frequently in cancers. Okay, so this is MAD2. This is this protein that came up in this paper we read the other week. And it activates the spindle checkpoint. And what it does is it binds to unattached kinetochores. And so here you can see your microtubules in green, your chromosomes in blue, and your microtubules are bound to your chromosomes. But look, you have a kinetochore here that's not attached. That chromosome should not be down there. It should be up here with the rest of them. So it's an unattached kinetochore, and it was bound by MAD2. And that will activate the spindle checkpoint. And that will result in inactivation of CDC20, which inactivates or prevents activation of APC. Yes?
It would also deactivate MCDK as well, right? You need a phosphorylate activation.
You would also inactivate MCDK. I don't know. So you said, why you said we would inactivate MCDK?
You need your APC to be phosphorylated, and then CDC20 binds to it and activates it.
Right. So normally you would need to phosphorylate CDC20, activate APC, and that's the normal progression. But here you're preventing activation of APC. But so really, MCDK is still active. You can't activate APC. You can't actually exit. Yeah.
I'm sorry. So when we activate the checkpoint, I know that we talked about all the things that like the proteins do and don't do, but in terms of the cell cycle, does this like target the cell for death, or does it just sit there, or like
So what this does, when you activate this checkpoint, it arrests cell cycle. And we were talking about this. We were talking about this in recitation today. Is what happens after that. One would expect that if your cell cycles are arrested for too long, it should result in something, right? Because you're not growing. Okay. And I guess the question would be, how does the activate apoptosis? There's got to be some connection between prolonged cell cycle arrest and apoptosis. Okay, so remember, CDC20 is required for activation of APC. So if you have no APC that's active, you have no anaphase. Right, because APC needs to degrade securin, which activates separase, which cleaves cohesin. But if all goes well, right, so if all goes well and your mitotic spindle attaches to kinetochores appropriately, then separase can cleave the cohesins because APC will be activated. So normally you've got securin bound to separase, which keeps it inactive. And interesting, it's showing another level of regulation. But as long as but once APC is active, securin is degraded and separase can be activated. So there's actually 3 mechanisms for keeping separase inactive until it's needed. One is it needs to be phosphorylated. 2 is normally it's bound by securin, which keeps it inactive. And then 3, there's this protein called shugoshin, which actually binds to and covers cohesins. So it's a second level of regulation. And shugoshin is degraded by APC. So APC activation is going to induce degradation of shugoshin and induce degradation of securin, which allows for APC. Induce degradation of securin, which allows for separase to cleave cohesins. Is it important to keep your sister chromatids together until we're sure everything's correct? Yeah, so it makes sense that you have multiple levels of regulation. Okay, so APC is activated towards the end of M phase, and that allows for appropriate sister chromatid segregation. Once the mitotic spindle is appropriately attached, and APC is activated and cohesins are cleaved, cells can proceed through anaphase. And so then you've got anaphase A and anaphase B. Anaphase A is when you've got this microtubules being shrinking, microtubules of the mitotic spindle shrinking, and so that's catastrophe of those microtubules. And so that happens. So you've got your kinetochore and your chromosome, and you've got your mitotic spindle bound, and you got that catastrophe of your mitotic spindle, and that force pulls apart your sister chromatids. That kinetochore kind of acts like a collar, so it's bound to the chromosome and bound to the mitotic spindle in such a way that even though you've got this rapid disassembly of your microtubules, the mitotic spindle stays attached to the chromosomes. So this allows the chromosome to be attached to the spindle despite this rapid depolymerization. So you get this movement. The catastrophe is controlling the movement of sister chromatids towards the spindle poles. So you've got that. So then you also have anaphase B. So that is anaphase A. Then you have anaphase B is the spindle poles being pushed apart. And so that's dependent upon motor proteins. And you've got these, so you remember we talked about these microtubules that slide against each other, and they've got these motor proteins with the 2 motor domains that both walk to the plus end. And so it's that activity that's going to slide them apart. So you've got 2 things, the shortening of your microtubules and then the pushing apart. of your spindle. And then you've got telophase. Your nuclear envelope reforms, your mitotic spindle disassembles, your chromosomes decondense. So, before we go, let me ask you, if you stained your cells at this point, at telophase, with PI, Propidium idae, and you did flow cytometry, what would the data look like? How much 1x would you have, and how much, would you have more 1x or more 2x?
You just exited from mitosis.
You're in telophase.
Oh, you have more to this.
More 2x, right? Because you haven't done cytokinesis yet. You still have twice as much DNA. And so you're going to have more. What if you wait a little bit longer? Would you have mostly 1x or mostly 2x? Mostly 1x, because then after this you're going to do cytokinesis. You'll be back to 1 copy of DNA per cell. Okay, that's it, that's all.